Hello students, this is chapter 19, excretory products and their elimination. And in this this is video number 2, and in this video, we'll be studying about urine formation. In the previous videos, I have discussed about the structure of kidney and nephron. If you all have not seen that, please go through that video. Now, what is urine? Urine is a liquid. It's a byproduct that is secreted by the kidney. And we all know that the purpose of formation of urine is to remove the waste like urea and uric acid and excess of water which is present in the blood. And in this urine formation, the kidneys, they undergo filtration. And we also know, children, that the basic structural and functional unit of kidney is nephron. Now, this kidney has millions of nephrons packed together, and the urine formation takes place in 3 steps: filtration, reabsorption, and secretion. Now, the very first step, that is filtration, or also known as ultrafiltration. Now, the blood, we all know that enters through afferent arteriole, you can see over here, and comes into the glomerulus and then comes out through the efferent arteriole. When it enters through afferent arteriole and comes in the glomerulus, the diameter of the afferent arteriole is more and the diameter of efferent arteriole is less. So there is a pressure buildup in the glomerulus over here. The pressure is built up over here in the glomerulus, and then the filtration takes place. This filtration is also known as glomerular filtration. The blood has to pass through 3 layers. It gets filtered through 3 layers. The very first, endothelium wall of the glomerulus, which is this pink layer, this pink. This is the endothelium of the endothelial cell of the glomerulus. Then you can see over here the maroon color layer. This is the basement membrane. Then it passes to the 3rd layer. You can see this blue color layers. This is the epithelial cell of Bowman's capsule. These epithelial cells of Bowman's capsule is known as podocyte because it's foot-like. It's something like this. The structure of these podocyte cells are something like this. And in between the podocyte cell, there is a slit, the filtration slit. Through this slit, the liquid passes and comes into this space, that is the Bowman's space over here. And from here, it goes into the tubule. Nearly 1100 to 1200 ml of blood is filtered by the kidney per minute. And the amount of filtrate formed by the kidney per minute is called, it is regulated by glomerular filtration rate, GFR. The second step is reabsorption. Almost this 99% of the filtrate is reabsorbed by the renal tubules. This is because it contains large amount of useful substances. These useful substances are reabsorbed by the epithelial cells of the tubules, and then it is sent back to the blood. And if you can see over here, see, the bicarbonate ion, the NaCl ion, the water, nutrients, potassium ion, they are all reabsorbed by the PCT. Talking about the loop of Henle, the descending loop of Henle reabsorbs water. It is permeable to water. But the descending loop of the ascending loop of Henle, it reabsorbs NaCl. And the DCT reabsorbs NaCl, water, and bicarbonate ions. So the cells of the tubule, the 3rd portion is secretion. The 3rd step is secretion. The cells of the tubule secrete substance into the filtrate. It is done to maintain the ionic and the acid-base balance of the body fluid. If we see to each part of the tubule, then PCT, selective secretion of H ion, ammonia, and potassium ion takes place. Loop of Henle, as I told you, the descending limb transports water, and ascending limb, active and passive transport of electrolyte takes place. And the distal convoluted tubule, the DCT, again selective secretion of H ion, ammonia, and potassium ion. And the collecting duct, there is a selective secretion of H ion and potassium ion. Now let's understand what is counter current mechanism. If you see only the loop of Henle, just focus on the loop of Henle, then, okay, let me change the color. See, the loop of Henle, this is the descending limb of loop of Henle, and this is the ascending limb of loop of Henle. Oh, the color is quite similar, so let me change it, make it green. Now, in these two limbs, the filtrate is flowing in opposite direction. In this limb, it is flowing in the downward direction, and in this limb, it is flowing in the upward direction. The peritubular capillary, this one, this is the peritubular capillary. If you see, this peritubular capillary also runs parallel to the loop of Henle over here. It also runs parallel to the loop of Henle. And the moment, if you take out this loop of this vasa recta and see, in vasa recta also, the blood is flowing in opposite direction in both the limbs. So it forms a counter current. The two limbs, they form a counter current. The two vasa recta forms a counter current, as well as the vasa recta and the loop of Henle also forms a counter current. So the filtrate flows in opposite direction through the loop of Henle, and therefore it forms a counter current. The descending limb, as I told you just now, is permeable to water. And so you can see over here, the water is reabsorbed from the descending limb. Now, when the water is reabsorbed, you can see the filter gets concentrated, filtrate gets concentrated. Here is 300 osmolarity, then 400, then 600, 800, and then 1200 at the center. Now, the ascending limb is permeable to electrolyte, whereas it is impermeable to water. So it starts reabsorbing NaCl, as you can see over here. It is reabsorbing NaCl, and as the fluid moves in the upward direction, the osmolarity again reduces, because now the NaCl moves out of the ascending limb. The proximity between the Henle's loop and vasa recta also, as I told you just now, forms a counter current, and this counter current helps in maintaining and increasing osmolarity towards the inner medullary interstitium. So you can even see over here, you can see here, the filtrate entering the descending limb and progressively more concentrated as it loses water. You can see it's coming 300, then 600, then 900 osmolarity, then 1200. Why? Because you can see over here the arrow, the water is going out. And where is it going? Into the vasa recta. And in this limb of vasa recta, the osmolarity is reducing. Now, when the filtrate is passing into the ascending limb is permeable to NaCl. You can see the arrow moving here. The NaCl is going out. And as the NaCl is moving out, the osmolarity is decreasing and the liquid is becoming dilute. So this counter current helps in maintaining the osmolarity. Thank you. And with this, we finish the second video. And very soon, I'll be bringing the third part of it. Thank you very much.